Welcome everybody to our webinar on antiperspirants and deodorants and innovation. This is a joint webinar between Cosmetics Cluster UK, of which I'm a director, and the National Biofilms Innovation Centre. We partner with them on topics that are relevant for research and innovation, which is one of our pillars for the Cosmetics Cluster. So it's very interesting for us to be involved. We've got a great lineup of speakers today, and also we've got attendees from various different backgrounds. So that's really exciting to see. We are recording, so I just got to see a message there. We are actually recording, so please don't worry too much if you're not going to take notes. There is going to be a recording available. So Katrina Steventon from NBIC and myself are going to start the proceedings, and we're going to give you some sort of an overview of the market and some pointers. And then Katrina will hand over to our first speaker. I suppose the first thing to say is the differences between antiperspirants and deodorants, which goes without saying, but they are different in function and in application. And deodorants are actually one of the most important personal care products that are used every day. So an antiperspirant works by controlling the sweating and body odor in two different steps, by firstly preventing the sweat reaching the skin surface, and secondly, it eliminates the bacteria that causes body odor. Whereas a deodorant is a personal hygiene product that's applied to the body to prevent odor. So that's the application side. In terms of the market, in 2019, it was estimated around $75 billion US. I'm sorry, I can't give you that in pounds, but it's in dollars, which is approximately 22% of the global personal care market of that year of $339 billion. So as you can see, it's about 20%, so about a fifth of the market, which is, not an insignificant amount. North America is the largest market, followed by Europe and then the Asia Pacific. Asia Pacific is expected to increasingly grow with the rise in disposable income in countries such as India and China. I have to say, our thoughts are going out to India at the moment. It's a terrible time for them, and hopefully, things will get back on track and they will be able to overcome the current crisis. In terms of the key players in the market for deodorants, we've got Unilever, Procter & Gamble, L'Oreal, Beiersdorf, all major global companies with significant contributions, with brands that we all will recognize, such as Dove. But what's increasingly happening is the emergence of natural and eco-led indie brands, the startup brands and the smaller brands, such as Modern Botany, who we have speaking today, Aurelia, Awake Organics, and I've seen recently a brand called Fussy, which has just come onto the market. The thing that the developments on the eco side seem to be very much around packaging and refillable packaging, but Katrina will touch on that in a minute. In terms of the geography and the product format, what we see is preferences in different parts of the world. Seem to be aerosol sprays and roll-ons in Europe, whereas sticks and gels in the United States. But again, that's been affected by some of the consumer preferences that are coming through. And one form that we're seeing more of is a cream. And I've mentioned refillable packaging. Well, Dove have just relaunched something in a refillable format very recently. So alternative delivery for aerosols is also important. So that's a quick tour around the market and the product types. Katrina is going to talk more specifically about the technology.
Good afternoon. I'm Katarina Steventon from the National Biofilms Innovation Centre. New opportunities can be either technology or consumer driven. The industry are constantly trying to improve the user experience as consumers demand more from their personal care products, be that in terms of product mildness, the skin feel, no sting, no stain, body responsive products that are quick to dry and obviously have good performance, long lasting effects. We see more of conscious consumerism where with natural products and the self-care, well-being, hygiene, safety, and kindness to self and the environment space is blending. People want refillable and recyclable packaging. And we understand the consumer concerns more, feeling self-conscious and isolated with body odor. The industry are wanting new selective antimicrobial technologies. The axilla is a great environment, a hotspot for malodor forming bacteria. And the R&D innovation focus is on inhibiting body odor. Deodorants work by either inhibiting or killing all underarm bacteria. And we feel there is an opportunity to create new deodorants that target specific species. So what is the future of deodorant technologies? Our understanding of the microbiology and of the biochemical origin of human axillary odor still needs more research. Biofilms in the axilla have not been confirmed. However, presence of clusters has been implied in some studies. We at NBIC aim to partner our academic expertise with industry developing new products, either new finished products or new ingredients and deodorizing technology. And we have commissioned an independent report looking at the commercial value of addressing biofilms in personal care. And the infographics you can see on the right-hand side are available for download on our website. We have a great lineup of speakers today. And it is my pleasure to introduce Deanna Utroske from the US. Deanna is a thought leader and a visionary, and she will talk about trends in deodorant innovation. Thank you.
Thank you, Katrina. That was a lovely introduction. For those folks who I haven't yet had the chance to meet, I'll just say again that my name is Deanna Utroske, and you might recognize me from a video series I do called DU Views, from my work with cosmeticsdesign.com, where I've been the editor for several years, or of course I've spoken at any number of presentations or moderated panels for beauty industry events around the world. If you could actually show the previous slide quickly. My email address is there. It's hello@deannautroske.com. And I will have a text version of my remarks today. So if you just want to send me a note, if that's of interest to you, I can share a PDF. But I'm so grateful that you're here. And I want to also say thank you to both the National Biofilms Innovation Centre and to Cosmetics Cluster UK for inviting me to speak as part of today's Innovation in Deodorants webinar. The second slide, thank you. So today I am speaking about innovations, improvements, and ideas for the next decade of deodorants and antiperspirants. In our modern moment, wellness, hygiene, and aroma are more central than ever, which means that there's a big opportunity here for deodorants and for antiperspirants to take on a new importance in the marketplace and to meet beauty consumer expectations head on. And that should finish the slides at the opening. Thank you. If we look at these overarching trends, you can hold that slide till the very close of my talk. I'm happy to speak to camera here for the remainder of my session. If we look at these overarching trends, wellness, personal hygiene, aromatherapy, and let's add self-care in here too. And if we then consider skincare and hair care as top performing categories through the pandemic, we start to get a sense of the sorts of deodorant innovations that will appeal to beauty consumers today and in the future. Now, I know we are all very eager for this to be a future where our professional lives and our personal lives are not spent reacting to the pandemic. But the months and years that it might take for us to truly get through this pandemic will undoubtedly influence consumer expectations and behavior for even longer than that term. So it's very much worth noticing how consumers and the industry responds to the pandemic. So be watching the emerging immunocosmetics movement and consider how it crosses over into deodorant, for instance, as triclosan, which may weaken the immune system when used in excess, falls out of favor. Pay attention to wellness and personal hygiene and aromatherapy trends. As we've heard mentioned already, pay attention to environmental sustainability and ethical business issues like fragrance ingredient transparency. And be aware of unexpected competition as well from popular brands like Hint. This is a beverage brand led by Cara Golden that now includes sun care and, as of January, a deodorant in its product portfolio. So that's a beverage brand. But besides the unexpected angle of this product launch, Hint made another wise move in their deodorant formulation in that it's made without what's known as a fairly on-trend ingredient, right? Coconut oil. But this ingredient also happens to be an allergen for some people. So as Katrina mentioned, as is often the case when we're looking for novel approaches to personal care, for next generation concepts, and for brands that really resonate with consumers, I want to move for a moment to look at what's happening in the startup and indie beauty space, so much as it includes deodorant products. Now, if you've heard or seen any of my previous content about deodorants, you have likely already heard of a brand called Sway, founded by Rebecca So in 2017. Sway has since expanded beyond underarm care, but it got its start with a 3-step skincare and deodorant routine for underarms, which is an indication that innovation in this category need not be limited to the most common product benefits. It's also important to recognize that a product as widely used as deodorant, what did we hear, 20 or 22% of the market, can be leveraged to effectively introduce consumers to an entirely new approach to beauty care formulation. It was some 5 years ago, I think, when Carmen Craig, who was at the time working for the brand Ursa Major, told me that, quote, deodorant is the gateway to natural personal care, close quote. So if an ingredient or a formulation strategy performs well in this category, in the deodorant category, it's infinitely easier, perhaps I should say, to sell it across the board. Smarty Pits is another brand that I've been watching for some time. Stacia Guzzo founded Smarty Pits to meet consumer demand for aluminum-free deodorants, but the brand is just as well known now for its prebiotic approach to deodorant formulation. And at this point in time, there are numerous brands in the microbiome deo space, but I want to mention one other here, and that's a brand called Kinko, a Texas-based brand that takes its name from the Japanese language, which I don't speak, but I understand that Kinko means balance. This brand has a fair amount of consumer education content about the underarm microbiome on its site, and Kinko centers its product formulas on a patent-pending combination of pre- and postbiotic ingredients that it calls microbiomix, which leads us nicely into a discussion of emerging ingredient technologies. Though, truth be told, I haven't seen many new deodorant-specific ingredient launches over the past year. One notable exception is from Vytrus Biotech. And I believe that an expert from this company is speaking later on in this webinar. So I'll just share here that the Vytrus ingredient Deobiome noni is based on prebiotic technology and is being referred to as a biological deodorant. Another relatively new deo ingredient is called CtraMild. This is from ICL, a specialty ingredient maker based in Israel. According to ICL promotional materials, the ingredient is a molecule sourced from Dead Sea minerals and, quote, works by controlling the odor-causing bacteria on the skin surface, close quote. It promises to support deodorant formulation without aluminum, without alcohol, without antibacterials or paraben. It also purports to provide sweat and sebum absorption and anti-stain protection, among other benefits. There's a fairly new brand called Pit Liquor from a couple called Erica and Jason in Colorado. I will not attempt to pronounce their last name, but if I share the PDF with you, you can Google them. They're seeing good results with the formula that they've created out of organic distilled vodka, local whiskey, and teas that have antimicrobial properties. So while some are saying that alcohol is on its way out as a deodorant ingredient, this brand, as well as carbon capture technology, which I'll mention again later on, seem to indicate otherwise. Italy-based cosmetic and pharma ingredient maker Calichem has an ingredient called DeoHapDry, which is a biomimetic hydroxyapatite that the company is promoting as an alternative to aluminum salts as a sweat control ingredient in deodorant formulations. And P&G's Secret brand launched Secret Derma Plus this past February, which is formulated with vitamin B5 and something that the company calls Derma Shield, a quote, proprietary and innovative combination of skin conditioning ingredients, close quote. So here we're seeing skincare influence formulation and benefit claims in this category. So if we're thinking now about relevant microbiome science, I want to read just a bit from a 2019 article published in the peer-reviewed journal called BCM Biology. The article is titled "The Impact of Skincare Products on Skin Chemistry and Microbiome Dynamics." Now, this article is the work of some 20 contributing authors, so I'll just name a couple here. The lead authors on the piece are Amina Buslimi and Ricardo da Silva. They both work at the Collaborative Mass Spectrometry Innovation Centre at the Skaggs School of Pharmacy and Pharmaceutical Sciences in San Diego, California. And I see folks asking about slides. There are no slides for this piece of the presentation. It's just my clever remarks that you're stuck with this morning. So I'll go ahead and quote now from that article. Mass spectrometry, they say, mass spectrometry and 16S rRNA inventories of the skin revealed decreases in chemical as well as in bacteria and archaeal diversity on halting deodorant use. Specific compounds from beauty practices used before the study remained detectable with half-lives of 0.5 to 1.9 weeks. The deodorant and foot powder increased molecular bacteria and archaeal diversity, while arm and face lotions had little effect on bacterial and archaeal, but increased chemical diversity. Personal care product effects last for weeks and produce highly individualized responses, including alterations in steroid and pheromone levels and in bacteria and archaeal ecosystem structure and dynamics. Close quote. So what's interesting here is that the easy assumption to make, right, for maybe a casual observer, is that personal care products, especially those formulated with conventional preservative ingredients, would seem to diminish microbial populations, and that's not the case. So while I'm not a scientist, and I certainly don't read all of the literature or research on the human skin microbiome, I am sure that we have infinitely more to learn when it comes to the microbiome. I am sure that even though highly accomplished teams at companies ranging from L'Oreal and IFF have been dedicated to learning about the human skin microbiome for some 10 years, if not longer, and applying that knowledge to skincare, scalp care, and body care, I'm still sure that we have more to learn, that new knowledge will be emerging for decades to come. Now, as we heard, I think Jane was noting different product formats for this category. It actually can be quite difficult, somewhat challenging to find something truly new. We have liquids, gels, solid stick deodorants, as well as deodorant bars. We have sprays, powders, paste, creams, crystals, balms, deodorant pads, deodorant wipes. There are even deodorant from within products, edible or ingestible candies and gums that are often formulated with geraniol that comes from roses. And they might more accurately be called, I suppose, fragrance from within products, but all the same, I suspect there is something to this deodorant from within concept, especially as supplements have seen a lift in consumer interest in recent months. Now, the array of deodorant formats to consider when developing a new innovative product isn't limited to a select few of those that I've mentioned. Many, if not all of these deo formats are doing well in the marketplace today and will continue to be go-to options for personal care consumers in the future. Act, for instance, a brand out of London, sells a deodorant balm in a squeezable aluminum tube and also sells a gua sha-like applicator meant to help with lymphatic drainage. Deodorant pastes like the one sold by Australia-based Black Chicken Remedies, are quite popular, particularly among natural beauty consumers. Ethique, the New Zealand-based brand, which is well known for its plastic-free solid beauty and personal care products, makes deodorant bars, as does the Singapore-based brand called Smood Natural Deodorants. Brands like Pacifica, EO, Wish, and LA Fresh all have deodorant wipes in their product portfolios. And near the start of 2019, Unilever got in on the deodorant wipe trend with wipes for use not only on underarms, but also the neck and chest. There are also brands creating customized and personalized deodorants. An example here is a Vancouver, Canada-based brand called Nala. And when we go ahead and look at the spectrum of deodorant packaging, we see everything. We see plastic, glass, paperboard, and even cylindrical tubes made of stainless steel, like the ones used by Nonico, a brand that gets its metal packaging from a company called Verity. There are deodorants in crushable aluminum tubes like ACT, which I just mentioned, and brands like Type A are putting deodorant in plastic tubes more commonly associated with sun care or body care products. There are refillable deodorant packages and some made of truly mono-material plastic that can be recycled. Now, I don't pretend that my knowledge of deodorants is exhaustive by any means, but a few things that I have not seen are deodorants really with the range of sensorial properties like we see in skincare. Deodorants that change phase or format upon dispensing or application, I haven't seen. I have not seen biphase deodorant, and I haven't seen deodorant pearls or beads or ampules. I also haven't seen like an in-shower, the way we do like a body exfoliator that leaves you moisturized when you exit the shower. I haven't seen a product like that that leaves you deodorized when you step out of the shower. So some potential there. And there are, of course, further opportunities in deodorants in terms of novel dispensing mechanisms or applicators, and certainly in terms of addressing expectations and benefits of diverse consumers, including the baby boomer generation and consumers in the bracket, the age bracket of over 40. Now, before I finish, I want to say a few things about odor and deodorant beyond the underarm, because there's always an opportunity, right, to transfer innovation across categories and to find inspiration in unlikely places. There's a new brand in the oral care space called Mouth Off that has launched a dissolving gum. This cleans the breath and is being marketed as a product rather that's more sustainable to similar products because it's easily ingestible and eliminates what would basically be post-consumer product waste, right? Which with the conventional mouthwash or gum requires that the used product actually be spit out. So this is a curious turn on sustainability. There's an essential oils educational platform called the Tirasand Institute, and they have an interesting post on their site about the potential of essential oils as deodorant ingredients and how the efficacy of various oils may depend on site-specific microbe populations. This post discusses the use of cypress oil, for instance, as an underarm deodorant ingredient, and then something like juniper berry oil as a deodorant for the feet. There is also potential for informed personal care product innovation as knowledge of the vaginal microbiome increases. Now, I say informed innovation because a recent item published in the expert review of Anti-Infective Therapy Journal, which Taylor and Francis puts out, explained that, quote, the mechanism by which some vaginal cleansing products cause health problems is linked to disruptions in the vaginal microbiome, and that vaginal deodorants have been shown to alter the vaginal microbiome over time, close quote. So I'll link you to this and other resources that I've mentioned in my PDF version of this talk, so you can look further into that if you're interested. A couple of other ideas I've been wondering about when it comes to odor and scent and microbes are odor pairing or encapsulation technologies like we see used in home care. I know also that several companies have developed and patented microbes, and I wonder if this could extend to a sort of biotherapeutic approach to deodorant, maybe editing microbes to off-gas pleasant scents and adding them to the skin. Now, I know it's I'm making it sound much more easy than it would be in practice, but I think it's still quite an intriguing concept. And I suspect that carbon capture technology, which I mentioned earlier, the kind of technology that we're seeing from companies like Lanza Tech, and that's L-A-N-Z-A Tech. I know folks in beauty are used to another Lanza. This company produces an ethanol that's made using captured carbon, and Coty is already planning to use it in their fragrance formulations. I think by 2023. really across the board. So I suspect this sort of technology will give new life really for at least the short term, right, to alcohols and other personal care product formulations, including deodorants. So I'll let you show the final slide for my talk now. And it's just to mention that, as promised, in the days ahead, I'm putting together a PDF of my remarks here. And that PDF will include links to brands, to the news I've mentioned, and other resources that I've pointed you to throughout this talk. If you'd like a copy of this Future Fresh, the Next Decade of Deodorants PDF, just drop me a note at hello@deannautroske.com. That's DEANNAUTROSKE.com, and I will get you that PDF. So with that said, again, I want to send my thanks out to the National Biofilms Innovation Centre and to Cosmetics Cluster UK for inviting me here for this talk today. I thank you all for watching, and I hope you do enjoy the rest of this Innovations in Deodorant webinar. Thanks so much.
Thank you very much, Deanna. Beautiful insights and new trends. I know you are not staying with us until the end, so if there are any questions for Deanna, please do type them in or unmute yourself to ask.
Thank you.
Everybody was mesmerized with So you can always follow up with us in terms of questions, and we will go on to the next speaker.
Wonderful.
Thank you, Deanna.
Thank you all.
It is my pleasure to invite the in vitro testing expert. So Dr. Carol Treasure is the founder and CEO of Accelerate, which is animal-free testing lab, and her company's innovation has been recognized at regulator level by funders, the Innovate UK and also Horizon 2020. So Carol, please, thank you for sharing and start your talk.
Hi, thank you very much. I hope everybody can hear me okay and see my slides. Yeah?
Yes.
Okay. So I'd like to just give a little bit of a whistle-stop tour with the time that we've got available today to think about the in vitro tests that are available to support the development of deodorants. And we know that deodorants contain both synthetic and natural ingredients that are capable, in some cases, of causing irritant or allergic contact dermatitis. So it's very important to understand a new deodorant formulation from the outset, not only from the perspective of the basic safety assessment, but also in terms of providing supportive data for claims in a world where, as we know, and Deanna mentioned, consumers are ever more keen on being assured that the formulations they're using are mild to the skin. So what I'd like to do is just take you through some of the work that we're doing in this area. And just before we start talking about the tests themselves, I wanted to raise this point, which is relevant to all of the tests that come up in the later slides. It's important to remember that most in vitro methods today still use animal-derived components such as serum, tissue extracts, and antibodies. The reasons for that are largely historical. So when the methods were first developed, some groups didn't really have any alternative, but that's different today. And Accelerate is unique in terms of being a contract lab that has an animal product-free or vegan approach to our testing. And we're collaborating with the Vegan Society to accredit the tests that we provide. The reason we do that is really twofold. It's driven by science, because if we eradicate animal-derived components from the in vitro tests, we can create a much more human-relevant system and get higher reproducibility of our results. It's also driven by ethics. As we know, the consumers and industry are increasingly demanding sustainable, ethical products. And if we're providing a vegan range of products, for example, they need to be tested in a vegan compliant way. I won't go into this scale today, but just to point you to our website, or you can ask me about it. I think we have to acknowledge that we can't currently provide every single test in a fully vegan compliant way. But what we're aiming to do is to get to that point over the next few years. And most of the tests that we currently provide, we would give an ethical assessment so that companies can decide how important it is to them to be fully vegan compliant in their testing and whether a particular test is going to suit their needs. So against that backdrop, let's consider why we needed an in vitro test to predict mildness of deodorants and other products. There was a study conducted in Europe a few years ago, and I was really astounded by some of these statistics, which do seem to be very robust information. But 19.3% of those surveyed said that they'd had a skin reaction to a personal care product within the last month that lasted more than 3 days, 31.8% within a year, and 51.7% at some point in their lives. And importantly, this leads to people to actually actively avoiding particular products or brands in the future. 37% of people said that they would avoid a skincare brand, for example, if they'd had a reaction to it. So there's a definite commercial implication and not just a safety implication and claim support issue around the need for these tests. So if we just take a look at the skin irritation test to start with, and I'd like to run through irritation and sensitization. The irritation is a simpler one to start with, and that's traditionally been tested in vitro using reconstructed 3D human skin models, which are grown at the air-liquid interface, and they're suitable for testing both ingredients and finished products, which are applied directly to the tissue surface to model real-life exposure. Now, the standard method, which is accepted by OECD, measures a single exposure time to give a yes/no answer. So in other words, is this item an irritant or a non-irritant? That's validated against traditional animal test data. But we believe that a much more sensitive approach is required for today's very mild cosmetic ingredients and formulations, because those tests were originally developed for much harsher industrial chemicals, for example. So we need to go much more sensitive with in vitro tests when we're looking at mild ingredients and products. So we developed, using this 3D skin technology, a test that we call XtraMild, and we have made the test much more sensitive over a longer period of time. So what we're doing, instead of having a single exposure time, which is done in the traditional test, we have a variety of exposure time points going up to 48 hours. We have relevant positive and negative controls for the test. And what we do is to measure the metabolic activity of the cells within those skin models as an indicator of cell damage. And we obtain what we call an ET50 value, which is the time taken to reduce the viability of the cells to 50% compared with an untreated control. And I think a really unique thing about the project that we were able to do here is that we were able to validate this in vitro test directly against human in vivo patch testing, which we undertook in collaboration with Q-Test. So this is just some sample data to show the kind of profile that we get when we apply different ingredients to the surface of the skin models over 48 hours. And by calculating the ET50, we can then place those into a rank order of irritation potential, whereby a longer ET50 time actually means that the ingredient is milder because it's taking longer to have any impact on the cells. So this is just a standard validation experiment that we did with 4 surfactants. We're able to rank those surfactants in the same order of irritation potential as was seen in the clinical studies, where we've got the clinical score on the bottom row of the table there. So the lower the clinical score and the higher the ET50 value, the milder is the ingredient. And we were very pleased with the level of correlation that we had with the in vivo patch tests, which helped us to start to develop a prediction model for unknown ingredients that we would test. Now, I'm showing you here some data for some face soaps and facial cleansers, which I know is a little bit off topic from today. But for this slide, I just wanted to demonstrate what kind of thing we can do with this type of testing. And we'd undertaken this particular in-depth study for these products, where we've obtained ET50 values and plotted them for a variety of facial soaps and facial cleansers. And this followed an article that had appeared in the Independent newspaper in the UK, and we took some of the products that were discussed. Now, it was interesting to see, to start with, that we get a wide variety of ET50 values and therefore mildness values across a product category, as you might expect. But some of the things that we could see is that, for example, there's not a direct correlation between the price of a product and how mild it is to the skin. There were some products that were actively claiming that they had credentials that made them a lot milder than other products, but they didn't necessarily perform that well in the test. And we were able to compare soaps and facial cleansers from within the same brand to see how mild those product types were to the skin. So I think that where this really is now extremely useful for companies who are conducting this test is to be able to plot their own ET50 value in the context of the reference values that we have, and to see where their formulation is stacking up against leading formulations in the industry. And we are now going through this process looking at deodorants, because this was a particular research project that we had funding for. And what I wanted to raise today is to say that we're now generating this kind of information for a range of deodorants. And we are actually looking for collaborations to help build that reference data set. So if any people listening to the webinar today are interested in working with us on that to gain some interesting information about their own products, then please do get in touch. This test has a variety of applications for different product groups, and clearly, as I've mentioned, deodorants are a very important part of that. And I think we need to recognise that armpit skin health and deodorant mildness is somewhat overlooked compared to other product groups within the industry. There's a lot that we know and a lot of background research that has been done on skincare, for example, for facial skin, but we don't have that level of knowledge yet about deodorants, and I think there's a real gap there that needs to be filled. So that's our work on skin irritation using the 3D skin models. But I also just wanted to use a few minutes just to talk about skin sensitization. This is very different from skin irritation, and many people do confuse the two. Skin irritation is a one-off reversible reaction in the skin, just basic damage to the skin cells. Skin sensitization, however, engages the immune system and creates a memory of that exposure, leading to a permanent condition. So once a consumer is sensitized to a cosmetic ingredient or product, they'll have that condition for life. So it's actually quite a serious endpoint to assess in product development. And we know that about 1 to 3% of the European population suffer from contact allergy to a cosmetic ingredient, and that equates to about 7.5 million people. We believe that the in vitro test can provide an ethical alternative to human trials, because we don't really want to be conducting skin sensitization tests on human volunteers when it could lead to a permanent reaction. So therefore, the in vitro test can act as a pre-screen. Now, we know that preservatives and fragrances, which are used in deodorants, are the most common causes of skin sensitization in personal care products. And it's important to note that this is true, of course, for both natural and synthetic ingredients. Natural never necessarily means that a product is safe. So if we look at the skin sensitization adverse outcome pathway, we can see that biologically in the skin, this pathway consists of a number of different key events, ranging from contact with skin proteins on the surface and binding to skin proteins, activating the keratinocytes in the epidermis. They then release inflammatory mediators which activate the immune system, first via the dendritic cells in the skin and then through to the circulating T cells. And when we look at in vitro tests, we have an opportunity to look at the detailed mechanisms of different points in the pathway. So we're able to obtain really detailed information about how an ingredient might cause a sensitization effect. We like to use a decision tree to help people to come to the right decision about which tests they need to conduct. There are tests available for all of the key events. However, it's not economically feasible and not necessarily scientifically justified to perform all of them on every occasion. And so what we'd normally recommend is to look at key events 1 and 2, which model binding to skin proteins and then activation of the keratinocytes, to build a picture of whether the test item is having an impact that could lead to skin sensitization. And that's in line with the OECD's regulatory approach, which recommends that you have at least 2 tests in agreement. So therefore, if the 2 tests both give a sensitizer result, you conclude that that item is a sensitizer, and the same for a non-sensitizer. But if you get 1 positive, 1 negative result, then that's inconclusive. So only then would we recommend going on to look at key event 3, which is the activation of the immune cells. Now, there are other tests available on a non-regulatory basis, which will also give supporting data regarding your products. And the nice thing about some of these new innovative tests that are starting to come through the pipeline now is that they not only tell you whether something's a sensitizer or not, so going back to that yes/no answer, they'll actually give you a potency prediction on whether it's a weak, moderate, or strong sensitizer, which is increasingly important, of course, for safety assessment. So I just wanted to show a quick example. This is skin sensitization testing for a cosmetic ingredient, and we've incubated the ingredient with the cell cultures that you can see on the right-hand side. And these are human epidermal keratinocytes, which are cultured in completely animal product-free conditions. We've grown those with a variety of concentrations of the cosmetic ingredient. And what you can see in the graph on the left-hand side is that as the concentration of ingredient increases, we're getting an increased signal coming from those cells. And that is linked to a transfected gene, which is an indicator of skin sensitization potential. And we've got a dose response effect, and the threshold has been crossed. So therefore, that ingredient is a sensitizer. We've also carried out the DPRA test for this ingredient, which measures the binding of the ingredient to skin proteins. And that also crossed the threshold to be classified as a sensitizer. So in this instance, we've got that 2 out of 3 classification that we need to conclude that this ingredient is in fact a skin sensitizer. This has been a little bit of a rush through some of the tests that are available, but I just wanted to conclude with a couple of points about why would we choose to use in vitro tests to assess deodorant ingredients and formulations. As I said at the beginning, and I really want to stress this, I think that the ethical advantages are so important. We often talk about the ethical advantages of using in vitro as opposed to animal tests, but actually they have ethical advantages over the human patch tests as well, especially, as I mentioned, when it comes to skin sensitization, which can be a permanent condition. We're able to really standardize the conditions in vitro, and we're able to drill down into the individual parts of the mechanism of that pathway. And so we can build up a detailed understanding of how the reaction's happening, and that can help us to avoid that in the future, can help us to look at different chemistries, different ingredients, different interactions when we're building a formulation to build something that isn't going to stimulate those particular parts of the sensitization pathway. As I mentioned, we're building a database of reference values for benchmarking purposes. And that's something that can be much more difficult to generate using in vivo human volunteers. And I think one of the other things that's really important to remember for in vitro testing is that they can also support that brand differentiation in terms of providing a positive message about using the latest test methods. So this moves beyond a basic not tested on animals to more of a positive message about, well, how do you test your products? Because even though consumers now have an expectation that products will not be tested on animals, and of course we can't do that in Europe and other global regions, they still want to know that those products are safe for them and their families and friends to use. So I think that using this, showcasing this technology and being able to say, well, this is how we do test products. This is scientifically advanced in its approach, and it's more predictive about human health than the animal tests ever were. And some of the companies we work with are really putting that message across to their consumers now in a positive way to showcase how products are tested to make sure that they're developing things that are safe. So I think that's all I wanted to say, and just wanted to thank you very much and thank the organisers for inviting me.
Thank you very much, Carol. There is a question for you, but I would like to move on to the technology and tackle the question in the questions and answers, if that's okay. Thank you. So it's my pleasure to introduce Danielle Robustillo from Vytrus Biotech, who will talk about biological approach to body odor. Could you please upload your slides, Danielle?
Yep, I'll share my slides. There you go. Okay, can you see my presentation now?
We can. You need to go onto screen. Thank you.
Can you see it now?
Yes, lovely. Thank you. Good.
All right, so thank you very much for inviting me to talk about our line of work. So just introduce myself really fast. I'm Daniel Robustillo from Vytrus Biotech. And obviously, most importantly than myself is the company that I represent. We are based in Barcelona, and we have right now 15 ingredients in the market, in the pharma and cosmetic markets. And today I'm going to present our line of work in the deodorants, in the cosmetics, and try to explain you a little bit what we do. So I'm going to talk about this beautiful plant and how we work with it, which is the noni plant, the Morinda citrifolia. And I'm going to talk about our approach, so how we pretend to tackle and interact with this generation of odor in our body, but doing it without interacting with our natural physiology in our skin. So I'm going to talk about the quorum sensing, the relationship between the plants and the quorum sensing, our strategy, which is the quorum quenching, this new mechanism of action, and a little bit about the biological activity and clinical testing that we did with our ingredient. So first of all, just to introduce the quorum sensing concept, which I'm sure it's well known for most of you. The quorum sensing is a mechanism of coordination or communication between the bacteria. We just come out with this mechanism, and we really thought we could take part of it and interact with it with our plants. And what we try to do with this product, what we propose here, is to interact with this coordination system, to hack, let's say, this coordination system. So quorum sensing is involved in many bacterial or even fungi interactions, such as the generation of niche factors, which are key in the adaptation of this environmental, which is our skin. Quorum sensing is also involved in the biofilm formation and also in the different coordination, inter and intra-species coordination with the bacteria. So what we as well look at it is the LuxR and LuxS system, which is a system that is mediating the generation of those molecules that the bacteria use to communicate to each other, the autoinducers or the hormones that are generating the bacteria and that are able as well to detect and at certain threshold, a certain quantity of those bacteria, of those molecules, they are able to do coordinated behavior. This looks as system. It's been found in different genders of bacteria such as Staphylococcus, Corynebacterium, Pseudomonas, and Cutibacterium. And here in this image, I represent a little bit how all those mechanisms work. So the looks R and looks S system play a key role in the synthesis of those hormones, recognition, and when it reaches a certain threshold of quantity of those molecules to do a coordinated response of the bacteria. Some of the coordinated mechanisms that are mediated by those molecules, those autoinducers, those hormones are bioluminescence. Actually, this was how this quorum sensing was discovered. In Vibrio cholerae, also conjugation, antibiotic production, biofilm formation, and niche factors. Here, because the topic is deodorants or antiperspirants, we are going to focus on these niche factors that play a key role in the generation of the body odor. So, to develop these applications in deodorants, focusing in how to interact with this quorum sensing, we focus in the niche factors, which play a key role in the adaptation on the different microbiota or bacteria species in our skin. Those niche factors are related to the production of those growth inhibitory substances, to the production of those microbial communication molecules, to the synthesis of degradative enzymes, lipases, or the acidification by organic acid release or fatty acid release on the medium. So honestly, and obviously, these niche factors play a key role in the generation of the body odor. This is well known, but just to get the introduction here in the middle of the presentation, actually, our sweat fresh, when it's secreted for the apocrine gland, it doesn't smell. It is really the bacteria in our body, some species of bacteria that degrade that sweat with this fat and protein content and generate the malodorous substances. But to do that, they need a coordination. And this coordination is mediated by these niche factors and this quorum sensing coordination or communication. So here is where we focus our work to interact with those microbial mechanisms to really avoid this activity that is gonna end up forming those malodorous substances. So really here, a simplification of what I just said, really to make it understandable. What we are gonna do and our approach to get this deodorant and this deodorancy is to interact with those bacteria generating the body odor, avoid this metabolization of the fresh sweat, avoiding this cluster formation in the armpit, and avoid the generation of those volatiles that are ending up generating the body odor. So just a quick introduction here, which is not really the topic, but with the noni plant, with the Morinda citrifolia, we have been working with many species of bacteria and even fungi, because really the potential of this interaction with the bacterial communication goes beyond the body odor or the deodorants. We have been working with Malassezia, Furfur, with Cutibacterium, Acne, Pseudomonas aeruginosa, Staphylococcus aureus, Corynebacterium striatum, and Epidermophytum flocosum, and other bacteria that we are working with, because really we are really focusing in all the mechanisms that are mediated by this quorum sensing, and we think the potential of the noni plant, the way that we work with, it's far beyond deodorants, but still we for now have in the market product for deodorancy and also anti-acne as well with this mechanism. So just to talk a little bit about the plant, the Morinda citrifolia, as I said, the noni plant, it's a well-studied plant. I mean, more than 150 bioactives are being isolated and studied of this plant, and it has many activities described. But specifically, we work with this plant through our technology. So we based our active ingredients in working with plant stem cells. With this technology, allow us to work with the plants in a different way and look for different responses that obviously have to be in the plant, but developing those strategies of the plant further to get high efficacy and really focusing what we are looking for. So just a quick introduction of what we do really. What we do is to culture plant stem cells, here represented in this circle, and make those stem cells react, but modulating the response to the stresses that we apply to those stem cell cultures. In this case, what we do with the stem cells of the Morinda citrifolia with the noni plant is to simulate a microbial infection. What we do is to simulate, aggregate deactivated membrane fragments to the stem cells to make those stem cells of the plant react, generate a defense. This is a natural mechanism of the plant. What we do is to optimize that response. In this case, what we do is to optimize the production of terpenes, polyphenols, and phytosterols. This is what we are aiming here with this treatment. And what we have seen is that with the production of those molecules, we are able to reduce this communication system, this quorum sensing of the bacteria, in this case, related to the generation of body odor. We block the receptors, we reduce the generation of autoinducers in the bacteria, and we don't allow those bacteria to have coordinated behaviors. So this is summarizing what we are going to do. We are going to inhibit this quorum sensing activity with our callus culture or the Morinda citrifolia or the noni, and also we add a prebiotic cocktail actually here. We add to this callus culture of Morinda citrifolia a mixture of maltodextrins really, because we want to do a double strategy that we call this biological deodorant. We want to avoid this coordination intra and interspecies to metabolize the sweat, the fresh sweat, and generate body odor, but also we want to introduce another substrate to the skin, which is going to be this maltodextrin, to shift the metabolism and make those bacteria metabolize the sugars and avoid this generation of malodorous volatiles or substances. It's really a double strategy that we are proposing here. So just the idea of what we want to achieve here, to reduce the generation of volatiles and malodorous substances through this double strategy, those plant-based anti-hormones, and also this prebiotic cocktail based on maltodextrins. So we have a certain number of in vitro tests to prove and to really reassure that we are modulating this communication between the bacteria, this quorum sensing, which are this test, which is the bacteriostatic effect and fungistatic effect. This is the first test that we do with all our bacteria that we are studying and related to this generation of body odor and other skin affections. So we do an inoculum, which is the first column here. We do a count of the different colonies, and if we left it 24 hours, we see that really the growth is exponential. But by adding to the mixture these noni-stem cells, we see really that we achieve this bacteriostatic effect. We stop the growth, this growth of the bacteria, but without killing them. So we have this bacteriostatic, fungistatic approach. We don't want to have a bactericidal effect. We just want to don't allow that bacteria to metabolize all the sweat and grow exponentially really when we sweat, to avoid this excessive generation of malodor. But really we want to have this microbiota imbalance in our armpit. This is the first step. Second step is really to test the anti-biofilm effect, which directly has a relationship with this quorum sensing coordination that we want to stop at this point. So the idea is a similar test than the bacteriostatic, but here we add a glass coupon in the medium, in the vial, really to analyze if those bacteria are in suspension and also if those bacteria are generating biofilm through these laser scanning confocal microscopy images. So we test those 4 species. Here I'm just going to share the Staphylococcus aureus and Corynebacterium striatum, but the results of the 4 species were similar in means of the efficacy. What we've seen is these results. This is the case of Staphylococcus aureus, really. Here in the first graphs in the right side, we see the population in suspension. And we see similar results than the bacteriostatic test that we did before, really. We see that with the active ingredient, we are able to stop the growth. But this bacteria is still alive in suspension in the vial. And the add-on of this antibiofilm effect really is how we are able to reduce the biofilm formation. Here we have the control with the Staphylococcus aureus, which relates to this image, the control image, where we have all those colonies and forming this biofilm in the glass coupons. When we add the noni stem cells treated, we see really that we are able to avoid this generation of biofilm 99%, but with still all those bacteria in suspension in the vial. So really, here is the first proof that we are having, we are blocking this quorum sensing communication basic in the generation of biofilm. These are the results, Staphylococcus aureus, and those are the results of the Corynebacterium striatum. The numbers are similar in means of the that we avoid the generation of the biofilm, and here you can see the images as well, how all those glass coupons are not showing this formation of the biofilm, while we keep the species or the bacteria in suspension alive with this bacteriostatic effect. And the last test in the biological activity that I want to share with you is a specific marker of the Lux S gene expression, really. Here we did it in Cutibacterium agnes, but what we've seen is that we are able to reduce the expression of this Lux gene 89%, so directly related to this generation of the hormones or the autoinducers that the bacteria use to communicate or coordinate through this quorum sensing strategy. Really, this is the final demonstration that really we are interacting with this mechanism of action. So, as for the clinical evaluation, then we did a sniff test, really, after 24 hours of the last application of the active ingredient, and after as well 3 days of pre-treatment, which we thought it was important really, because here what we are proposing is a biological approach. We are proposing to have all our microbiota in our armpits getting influence in their physiology or metabolism. So really we thought 3 days of pre-treatment were necessary to really get to the point where we could have the maximum effect. So what we saw is that with our noni stem cells, we were able to reduce 30% the body odor. This is a deodorant, not an antiperspirant. Actually, we tested as an antiperspirant and we didn't see any significant difference, which is actually what we wanted to see in means of we want the skin to have their natural physiology and natural procedures. We just don't want that bacteria to increase the body odor or produce this body odor or malodorous substances. And to check that, we also did the gas chromatography plus a mass spectrometry to really measure the amount of volatiles and which volatiles we were having in our armpit, really, and what was the effect of our ingredient. What we've seen is that we are able to reduce 39% the total content of those compounds analyzed, malodorous compounds, and in more detail, and this is just one snapshot, we have the detail of all the molecules analyzed on those families, but what we've seen is that we are able to reduce, this 39% is the average, but we were able to reduce alcohols, 34%, aldehydes, aromatic alcohols, lactones, organic acids, sulfur-containing terpenes, really, in this gas chromatography. So really significant reduction on the production of those malodorous substances. Here, just a matter of the power of the image I show with you the gas chromatography, really, where you see really that the quantity of those volatiles is reduced significantly. It's just the image, but really don't treat it, and the treated gas chromatography profile differs significantly. So this is our line of work, what we have been doing with the deodorants, and really with this noni platform that we think that has a lot to say with this quorum sensing and microbiome interaction. So yeah, thanks. Thanks again to the organization, and yeah, feel free to ask any questions you may have. Thanks, Katrina, all yours.
Thank you, Daniel. We will leave the question until the end. It's my pleasure to invite the last speaker. He will speak about finished products. So Dr. Simon Jackson, who is CSO and co-founder of Modern Botany, and really an expert in pharmacognosy. And his approach, I find it refreshing. So he will give us insights into formulating a deodorant. Simon, could you please start your presentation?
Yeah, absolutely. Can you hear me okay, Katrina?
Yes, we can. Thank you.
Great. Okay. Can you see my screen?
Yes, we can see your screen as well.
Great. So thank you very much, everybody. And there's been some fantastic talks today. So I really appreciate you putting this together. So thank you, Jane at Cosmetic Clusters UK and also Katrina at National Biofilms Innovation Centre as well. So I really appreciate it. So a bit about myself. I'm a pharmacognosist. Nobody's ever heard of it, so don't worry. I'm going to explain what that means in a second. I'm also the Chief Scientific Officer and co-founder of Modern Botany. So today, I'm just very briefly in the time that we've got is I'll explain a bit about what is pharmacognosy. I'll give you a bit of background as to my career and where I came from. And also, is natural product science still relevant? I mean, I really was excited to hear about the noni that you just talked about, because it's great to hear that these natural ingredients are still alive and kicking. I'll explain a bit about natural deodorant formulations. I'll explain the bioactives and active ingredients that we're using, and maybe just explain the difference between synthetic and natural deodorants in the sector. And I'll give you a high-level overview of the kind of clinical testing that we've been doing and have done, and what we're planning on doing next steps, and a bit of a conclusion. So hopefully I'll cover all of that in the next 10 minutes. So pharmacognosy, it is a real discipline. It's normally found in a pharmacy college. It's derived from the Greek word, and a lot of Western medicine or allopathic medicine is derived from Greek medicine in the past. So pharmakon is drug or medicine, and gnosis is knowledge. So it's a knowledge of drugs or knowledge of medicine, and that's kind of what I specialize in. So pharmacognosy, it's a multidisciplinary study, and I'm not going to go into all the detail, but a lot of people ask me, what do you do? So I do a little bit of botany, a little bit of ethnobotany, which is traditional uses of plants, a bit about medical anthropology, which is traditional uses medicinally. Marine biology is very popular at the moment, microbiology, herbal medicine, then going into the chemistry, the phytochemistry, and also biotechnology, which is growing the ingredients. We also do pharmacology, which is the testing of ingredients on living tissue, and then also pharmaceutics, which is actually making it in large scale. And then you go into clinical pharmacy and pharmacy practice, which is the distribution chain. So it's the whole life cycle, basically, from a traditional use of a plant all the way through to discovering active ingredients to actually producing it as a medicine or a drug. So that's what I do. Is the study of pharmacognosy still relevant? I should think so, but I think at the moment everybody's very interested in natural ingredients. So for me, I really see it as the golden age of pharmacognosy, and it's really kind of starting to kick in, which is great. What I do is really just kind of keep the knowledge alive. So I'm trying to do a knowledge transfer to businesses and industry. It's about commercializing best practice and good manufacturing practice. It's also about making really good quality products. So for me, it's all about, being as good as your last product that you've made and showing the efficacy. It's also about benefit sharing. So if you're using a plant from a particular part of the world, it's about putting money back into that country of origin. So it's about how can we help rural communities with the kind of plants that we're using. So for me, I really do think that pharmacognosy is more relevant than ever, and obviously the deodorant sector is something that I'm very interested in. So myself, I am a doctor of pharmacognosy. I got my PhD at King's College in London. I specialised in skin cancer research. So that's my area of expertise. I did a postdoc at Kew Gardens. I have a big laboratory there called the Jodrell in Kew, and I also worked at GlaxoSmithKline because I used to have a big natural product division there as well, and I specialised in skin diseases. I was the CEO and founder of my namesake brand, Dr. Jackson's Natural Products, and I left that in 2016, and I'm now the Chief Scientific Officer of Modern Botany, which has been so since 2016. I'm also a member and guest editor of the American Botanic Council, and I'll come back to that later. Also the European Society for Plant Medicine, and a member of the Society of Cosmetic Scientists over in the UK as well. I'm not going to go through a whole life story here, but I am British, you can probably tell by my accent, but I've also lived in Southeast Asia, so I know the noni plant, for example, very well. And I've also been on expeditions to the Amazon, so I've worked a lot in Peru with the American Botanic Council, and more recently I've been working in Africa. reason why I've got this picture of the world is if you look at a map of the World Health Organization states, and the statistic is 80% of the world's population use plants as a primary healthcare. There's only 20% of the world's population that use allopathic medicine. So it's very much, I see, where we should be looking for new ingredients or future ingredients. So if anybody wants me to bore them with all my expeditions, I can do that another time. So we set up Modern Botany in 2016, and it's really about utilizing all of that experience from different expeditions and meeting with different healers and shamans and medicine men from around the world. And we've set up this, which is about the science of nature. So there's 3 of us in the company at the moment. There's myself, who provides the science side. There's Tracy Woodward, who is the new projects director, and you've also got John Murray, who's a sustainability director as well. So between the 3 of us, we've set up this called Soil, which is our key pillars of the business. So S is for science, O is for origin, I is for integrity of ingredients, and L is for longevity of people, but also for the planet as well. And this is really the core of all of our products and the business that we stand for. And we're very much about having the best soil that's available to provide the best ingredients. So you've got really the kind of top-notch ingredients. We do a natural deodorant, which launched in 2018, and we also do a multi-use oil as well. But our purpose is really more about education, and it's about educating people into the power of plants. And interestingly, the lady previously talking about natural doesn't always equal safe. And I think that's one of the key things that we're trying to get across to people. Just because you're using something natural doesn't actually mean that it's going to be a safe ingredient. We use what's called the power of 5. So these are my Cinderella species. So the species of plants that have been overlooked a lot. You've got flaxseed, you've got borage, you've got seaweed, you've got calendula, and you've got chamomile. I'll explain the reasons why we kind of chose those kind of key 5 ingredients. We're also vegan as a company, and we're cruelty-free international. One of the first companies. We're based in Ireland, by the way. We're also 1% for the planet, so we give 1% of our gross sales to environmental causes. We're also Cosmos Natural and obviously vegetarian approved as well. And we're en route to become B Corp registered in the near future. So my definition of success, it's not so much about the work that we do, but it's about the people that we help and the communities that we build. So that's really what we're doing in this business, is about supporting people through education, but also rural communities with building a good solid base. This is one of our products, which is a multi-use oil. It's been very much awarded from lots of different categories, but the deodorant is what we're going to talk about today. We've had a lot of awards on the deodorant since we launched, and I think a couple of years ago we got the Women's Health Best Deodorant Product, and last year we got the Most Innovative Irish Brand from Image Magazine as well. So I'll explain a bit about Modern Botany deodorant in a second. I know we've been talking about different stats, but the European and US cosmetic market is huge. You're talking 135 billion by the end of this year. And 10% of that is natural and organic beauty market, which again is about 11 billion US dollars. So it's huge, and it's looking to double by 2024. And if anything goes by what we've had in the last 12 months, our sales have gone up by 600%. So there's been massive increase in sales in natural products. It's definitely the fastest growing sector in the cosmetics industry. And I think the deodorant and personal care sector is one of the biggest in that as well. Consumers obviously very interested in more insights about what products are made of, especially with links between different specific cosmetic ingredients and health matters. So people are becoming a lot more aware as to what they're actually putting on their body. Interesting that you started out at the beginning with deodorant and antiperspirant. Do people know the difference? It's a question that I have to do quite a lot, because a lot of people don't actually understand the difference between deodorant and antiperspirant. Triclosan is the standard synthetic route for an antibacterial, and cyclomethacones are used as a solvent to get the triclosan to the actual site of action. And interesting what the guy was saying before, the sweat doesn't smell. It's the actual bacteria that feed off the sweat that produce the malodor or the body odor. So what I did is I looked at synthetic deodorants and said, well, we can get rid of the triclosan because there's a lot of really good natural antibacterial compounds. And that's what we did there. We got rid of the triclosan. And then the cyclomethicone, again, there's many solvents that we can use to actually get the active ingredient to site. So that's what I did with the basic formulation. From an antiperspirant point of view, this is to stop the sweating. Aluminium salts are used a lot, and aluminium chlorohydrate, aluminium formate are just standard ones that are used to block the pore. So again, just blocking the pore actually stops it from sweating, but is that healthy? You know, you do need to sweat, and we're built to actually sweat. So what we did is we got rid of the aluminium salts and we used a very strong astringent to actually tighten the pores and actually reduce the amount of sweat that people are having. So with regards to ingredients, one of the traditional ingredients that are used a lot is sandalwood from India and also Southeast Asia. You get a lot of sandalwood soaps. It's a very kind of woody floral scent. But the active ingredient in sandalwood is something called alpha-santalol. And alpha-santalol is a very strong antibacterial ingredient. So it's a very useful ingredient to use in a deodorant product. Lavender, we've all heard of lavender, lavandula angustifolia. Lavender, again, has got many interesting compounds, but mainly linalool, linalool acetate, and eucalyptol, all of them incredibly strong antibacterials and antimicrobial actives as well. And these slides will be shared at the end of the talk as well. Lemon tea tree is an interesting one from Australia, Leptospermum petersonii. Again, lovely fresh lemony smell, but really it's the geraniol and the nerol, which are the very strong antibacterial compounds in that. Frankincense, a lot of people don't realize, but frankincense is a strong antibacterial. Back in the biblical times, frankincense was very valuable because if you cut yourself before antibiotics, you could get septicemia really quickly. Frankincense is a resin. You cut the tree and the resin is then steam distilled. But again, frankincense has got some fantastic antibacterial compounds, and it is a medicinal plant. So you've got alpha-pinene compounds, which are very strong anti-inflammatory and antibacterial. Clove leaf extract, again, you've got eugenol, which is a very strong antiseptic and also an anesthetic. Coriander is a really interesting one. Coriander sativum is a very strong antibacterial, and it's actually used a lot as a preservative system in the industry. And it's very good for gram-positive and gram-negative bacteria. Sage, Salvia officinalis, again, another medicinal plant. You've got Roman chamomile, which I'm very interested in because we're actually growing the Roman chamomile down here. You've got apigenin, alpha bisabolol, and the blue chamazoline, which you get in the German chamomile as well. So all of these are very strong antibacterials. Vetiver is from Southeast Asia. It's the root of a grass. And again, vetiver is used a lot in the perfume industry, but also in kind of deodorant products. So we use a vetiver extract, which has antifungal and antimicrobial activities. And then the last two is eucalyptus, which has a lovely fresh smell, and it's used a lot in cleansing products and cleaning products. But eucalyptol obviously is a very strong antibacterial ingredient. So Atlantic cedarwood, the last of our blend, is a really lovely woody smell, but again, it's got really strong antibacterial compounds. And then with regards to the antiseptic, antiperspirant, we use witch hazel. So a lot of people don't know what witch hazel looks like, but this is actually the flower of witch hazel. It comes out in about January time. It's a beautiful yellow flower, but it's the barks of the tree which actually is used to make the witch hazel water. And I don't know if a lot of people know, but it's traditionally from North America, and it's the Native American Cherokee who used to use it used to use it for cuts and grazes. So basically, if you'd cut yourself as an astringent, it would tighten the skin and it would stop any bleeding and it kind of make the wound kind of join back together again. You've probably all heard of a pharmacist called pharmacist Tilden Pond, or you might have heard of his skin cream called Pond Skin Cream. But Pond's skin cream was launched in the 1800s to much success, big global success, as a vanishing cream. Again, because of the astringent capabilities, it tightened all the pores. So if you had any blackheads, they'd all be gone, because basically it was tightening all the pores and pushing out all the kind of dirt in your skin. So it's a very strong astringent, and actually in America the FDA class it as a grass astringent agent. So it is actually listed as a medical astringent, and this is what we use really as our solvent to actually close the pores and get the product to the skin area. So that's witch hazel. The active ingredient that's in witch hazel is actually a tannin. You've probably all had a glass of red wine when your mouth feels very dry after a glass of red wine, and that's the tannins. It's a very drying agent, but it's hammermelatonin which is responsible. It's quite a tongue twister. Hammermelatonin is the actual active ingredient that we're looking at, and that's a flavanol. So what we've been doing over here in Ireland is testing some very simple antibacterial using our deodorant extract, and this is some agar plates just showing different colonies of different bacteria, and you can probably notice these 3 agar plates at the bottom which have actually got the deodorant extract in them, and you can see there's no colonies at all growing in the actual bacterial. So we're getting some very good indicators that we've got a very strong antibacterial ingredient that we're using in our deodorant product. And then more recently, been working with the Munster University, and we've been looking at chamomile as an ingredient, and actually looking at the sustainability of it, and if we can actually grow it as an ingredient over here. And what we're actually finding is, if you just look at this graph here, this is a commercial sample of chamomile that we've been growing, which is a very strong antibacterial product. But these are the Irish grown chamomile extracts, and you can see the yield of the oil is about 3 or 4 times higher than actually what we're finding from a commercial extract. So we're getting some really good quality extracts as well that we're putting into our products. Again, just a very simple indicator, German chamomile, which is a strong antibacterial, is bright blue. It's a petrol blue. It's a standard indicative color. If you look at the commercial sample on the left, it's a very mucky blue. And normally that's from solvent extraction, or it might have been mixed with a different plant that's not been identified properly. But if you look at the Irish essential oil that we've extracted, you can see it's bright blue. So we're getting some fantastic results, and the quality of the chamomile extracts are actually being analysed as we speak. This is a simple steam distillation quality control test that you can do, and you can see on the left you've got very little of this blue chamomile being extracted, but on the right hand side, using the Irish flowers that we've grown and dried, you've got a very high concentration of it. So we're actually trying to solve the problem of getting a low quality ingredient in our deodorant products by actually growing it ourselves. We're also finding that adulteration is a big issue. Feverfew, I don't know if people know, but feverfew basically looks like a chamomile. It can be adulterated into a chamomile extract. So what we've been doing is actually using these monographs with the American Botanic Council to actually identify the difference between chamomile and feverfew. And you can look at microscopical and macroscopically, and also you can look through TLC and HBTLC to actually identify the active ingredients that you're really kind of interested in. So hopefully, just in summary, I've explained a little bit about what a pharmacologist is, so hopefully you understand what that is now. And I very much see it, it's very important at the moment, and I think a lot of companies you could really do with kind of, the advice of kind of pharmacognosy, especially in the personal care sector. I just wanted to kind of explain a bit about my background, where I came from, and also to talk about the ingredients that we've used and why we've used them, really. So, so hopefully people have found that interesting. This is where we're based. I'm actually calling you from my garden studio at the moment, which is just here on the left. We're actually growing fields of chamomile. So if anybody's in the Irish area, please come and visit us. And you can see Ireland is the island of the Green Island. But if you look at Dublin is up on the east coast here, we're about 6-hour drive away from Dublin at the very bottom. And I'll just take you straight down. We're right down here. The next stop is America. But if anybody does fancy a trip to Ireland, you're more than welcome. And I'll just leave you my email address here, and my company website address is modernbotany.com. So so thank you very much, Katrina, and I hope that was in time.
Thank you very much, Simon. I believe we have few minutes for a Q&A session. So would you like, could I invite the speakers to unmute themselves and just come on? Okay, thank you. So we have taken you through from the trends to testing to ingredients and then to finally the finished products. And I believe there are some questions which I'll just read out. So the first one was for Carol. Referring to the essay, the viability essay on your third slide, the in vitro assays, how do they account for skin barrier? Do you mimic its presence in the in vitro assays or tweak with the concentrations, assuming a certain level of permeability? Could you please answer that?
Sure. The skin barrier is modelled in the three-dimensional reconstructed tissues that we're using. It's known that the in vitro skin models have a slightly thinner barrier in general than in vivo human skin, but they do mimic the lipid profile very well. What this means is that, if anything, what we would expect to see is increased absorption into the skin in vitro because the barrier is slightly thinner. From a safety perspective, that's good because it means that we will overestimate any effect that might be seen in terms of skin irritation. From a commercial perspective, that needs to be taken into account because we have to recognize that we may be overestimating any toxicity. However, in this test, because we validated our in vitro method directly against human in vivo patch tests, and we got a very strong correlation with the lab that was testing in vivo. So for this particular test and with the ingredients that we've used to validate it so far, it hasn't been an issue. I think it's something that we have to always keep in mind, bearing in mind the chemistry of different ingredients that we might want to test and how we might expect those to go through the skin barrier.
Lovely, thank you very much. And two questions for you, Daniel. How does quorum sensing management cope with inter-individual variability of the microbiome? It's the first one from Jill Westgate.
Yeah, well, here we have been focusing in the control of the affections that we are mediating with. So really to go deeper in this inter-individual variation, we should take more analysis in the RNA of the different phylums and do a deeper study in the volunteers at the end. But we first have been focusing in the different species, just measuring by the stripping test. So I guess I don't have the full answer of that because we should be testing it in the future.
Thank you. And the other below is interesting to see how you use GCMS. I'm wondering how you sample the body odor for your tests.
Oh, well, when you work with gas chromatography and mass spectrometry with to measure the body odor, what it's done is to take the pad that was in the armpit for the analysis, you put that in a bag with nitrogen, then you stay for 30 minutes, then you take a sample of this air, of this nitrogen with the volatiles, and then you analyze the volatiles actually. That's what you do, how you do it.
Okay, I have a Simon asking Carol, so I will allow you to ask directly. It is about China and testing.
Sure. Carol, I was just really excited because, as you know, in the cosmetics industry, if you sell anything in China, they basically test all of your products on animal tissue or animal tests. And obviously I can see huge opportunities for Accelerate, if that's something that you might be thinking of in the future, to kind of introduce the kind of testing you're doing by way of you know, regulating testing for products overseas, maybe. So it wasn't really a question, it was just more of a comment, I guess.
Yeah, it is exciting, and I think that what we've seen coming from China over the last few months especially is a series of announcements that they are really starting to open up to these new technologies, and they're very interested from the perspective of the scientific advantages and how relevant they are to human skin. And I think that there is that strong bias towards technology in China, whereas maybe they might have a slightly different take on some of the ethical aspects, but they're very keen on that scientific slant. And I think that certainly if we can work with the authorities there to build their confidence in these new methods, then that's an important point. And also, I think they are very keen to make sure that they have trained scientists within China who can perform these methods, because they don't want to switch away from their current tests until they have the capacity to perform the new ones, which is understandable.
Can I just make a comment there? The Cosmetics Cluster UK is a member of the Global Cosmetic Cluster, and we've actually launched officially this week the new sort of legal entity of the Global Cluster. And one of the members is actually China Botany, which is representing the Chinese. So one of the things that we're very keen on is to sort of facilitate communication between the two, obviously with China and everybody else, which we feel is very important. So if anybody has any thoughts on that, or things that they would like to be communicated, then I think, please let me know, because we want to sort of look at what we can do in terms of education and connection and building a bit of a bridge. So I thought that was appropriate to mention that based on that question.
Sounds great.
Thank you, Jane. That's great news as well. So Simon, you had one more question for Daniel about the activity of the noni. Would you like to ask?
Yeah, again, sorry, it's just more about the phytochemistry really, and I guess it's still quite early stages, but have you found with the noni extract, do you think it might be an active ingredient in the noni extract or ingredients, or do you think it might be like the whole extract? I mean, I don't know if it's used traditionally as I say, a deodorizer or antiperspirant, but what I've found in the past is it's normally the synergy of all the ingredients working together and basically mimicking what people do traditionally is normally what happens.
Yeah, really our way of work is to base our efficacy in the synergy. So what we base our strategies to work with the stem cells and enrich different fractions, but we end up using the whole thing. So we actually compare just purifying some of the molecules, and we found that really with the synergies where we make the difference, really with the stem cells or even with the extracts. I think you know better than me, obviously, but we based in our efficacy in the synergy mainly, but enriching certain groups of molecules through different stress processes.
Because one of the things that I've done previously is like a bioassay guided fractionation, where you actually fractionate the raw material and to find the active compounds or fractions. But I think in this case, it's probably best just to use the whole extract, but maybe mimic how the extract, I don't know if it's an alcohol extract or water extract, but maybe mimic how it's done traditionally.
Yeah, definitely. We had many tests with the purified molecule or even with certain groups of molecules compared with the whole thing, and the synergy shows to be much more efficacy than the single compounds, for sure.
Great.
Great, lovely. And last question from Mark to Daniel. Could these species become tolerant to this intervention with prolonged time?
Well, in fact, the quorum sensing mediated responses is one of the reasons why the bacteria get tolerant to all those treatments. So if you are able to interact with the quorum sensing of it, you are going to be able to avoid this tolerance gain. So I wouldn't be like redundant on it, but obviously we expect that interacting with the quorum sensing, we can avoid this building of the tolerance.
Lovely, thank you very much. I think that concludes questions. If somebody else wants to ask, please email us. So I'll just reiterate that we at NBIC help companies to find the right academic expertise. In this case, it's the axillary microbiome expertise. So if you are a company either developing new deodorants or formulating new deodorizing technologies, please come in touch.
I really found this afternoon's talks really fascinating, and it's one of the great advantages of partnerships that we at CCUK can pull together this kind of lineup, working with NBIC. So just thank you, everybody. Thank you to the speakers, and thanks to everybody that's attended. And we'll be doing more webinars in the future, so hopefully there will be another topic that we can jointly work on. So thanks, everyone.
Thank you very much, Jane. So we will be writing this up as a blog and the recording will be available to all of you. Any questions, as Natasha has put, just link, come back to us at NBIC email address. So that's goodbye from me. If the speakers want to stay behind, please do. Goodbye, everybody.
Bye-bye, take care, thanks.
Bye.
Bye, everyone.